Hi everybody. Today we shall discuss some of the questions from regulation of gene expression of the chapter molecular basis of inheritance. So you can have a view at the black operon and its regulation. In the absence of inducer or lactose or allolactose, and in the presence of inducer lactose or allolactose. So please record this diagram in your brain before answering the questions that are about to come. Let us straight away enter into the first question. Choose the incorrect statement. In eukaryotes, gene expression can be regulated at a translation level. So it can be regulated at translation level, various stages of transcription level. Gene expression is regulated by environmental conditions. So gene expression can also be regulated by environmental conditions besides the physiological and metabolic conditions or biochemical conditions. Bacteria use glucose as a direct source of energy. Yes, they can use the glucose as a direct source of energy in their respiration. Right? Bacteria can directly use lactose as source of energy. No, bacteria cannot utilize lactose as a direct source of energy. It has to be first absorbed and later it has to be hydrolyzed to glucose and galactose. And glucose can be directly used, and galactose, after being converted to glucose, can be used as source of energy. So lactose cannot be directly used as source of energy. Fourth option must be the correct one. Let us see the answer. Fourth one. Here is the explanation for it. Bacteria cannot utilize lactose as direct source of energy. It has to be hydrolyzed to glucose and galactose. And galactose has to be converted to glucose to use as energy source. In eukaryotes, gene expression is regulated at a transcription level, that is transcription process level, RNA processing level. So that already has studied that heterogeneous nuclear RNA is converted to active messenger RNA due to processing. Messenger RNA transport level, yes. There must be the transport of messenger RNA into cytoplasm from nucleus, and the translation level 2. Gene expression is regulated by physiological, metabolic, and environmental conditions. Bacteria can use glucose as a direct source of energy, as the genes for glucose metabolism are constitutive. Yes, because genes for glucose metabolism means those genes that are involved in glycolysis and other respiratory reactions and reactions. Right? They are constitutive. All the time they are expressed. Coming to the next question. The matching type. Constitutive gene. What do you mean by gene that is always expressed? And RNA and proteins are formed. Respective messenger RNA and proteins are formed. Are referred as constitutive genes. In this gene, I is the constitutive. So it is not regulated gene. It is a regulator gene, but not regulated gene. It is a constitutive gene. A3. Region where the activators bind to operon. The activators bind to operon at the promoter region. At the promoter region only activators bind. Right? So these activators are positive regulators. One of the accessory proteins which can bind to the promoter sequences and enhance the binding of RNA polymerase. So they are bind to promoter, particularly in case of lacoperon, they are bound to the activators. Right? Next one. Region where the repressors bind to the operon. Repressors bind to the operon at operator. It's C2. Regulated gene means gene whose expression is regulated. They are gene Z. gene Z, gene Y, gene A. All these three structural genes present together and transcribed together into single messenger RNA known as polycistronic messenger RNA is a regulated gene. So D1, what are options? A, 3, B, 4, C, 2, D1. So second option must be the correct one. Let us see the answer. Second one is given. Constitutive gene, gene I, that is region where the activators bind to operon, lack promoter, a promoter. Regulated gene, gene Z, in case of lack operon. Region where the repressors bind to the operon, operator. Coming to the next question. In prokaryotes, which is the predominant stage for regulation of gene expression? That is transcription initiation. It is directly given in NCERT book. In prokaryotes, gene expression is regulated mainly at a transcription initiation level. Means RNA polymerase binding region and its progress into the structural gene. The transcription initiation is correct, not a transcription elongation. Once the transcription is initiated, elongation is carried out by the core enzyme. Transcription termination. Transcription termination is regulated by the terminator sequences as well as a factor known as rho factor. Translation initiation. Translation initiation is regulated by initiation factor and the availability of initiator transfer RNA. Right? So, fourth option must be the correct one. Let us see the answer. Fourth one. In prokaryotes, control of the rate of transcription initiation is the predominant site for control of gene expression. It is controlled by inducer, repressor, activator, co-repressor, etc. If it is controlled by inducer, and if it is transcription is promoted, initiated by inducer, it is positive regulation. Right? If it is by repressor, negative regulation. Right? So activator, inducible operon, inducers. Activator, positive regulation. Inducers, inducible operon. So co-repressors and all that repressible one. Right? Coming next. Transcription elongation requires only core RNA polymerase. Transcription is terminated by rho factor or termination sequences. Translation initiation is regulated by initiation factors and availability of GTP and initiator amino acid transfer RNA. Coming to the next question. Two statements are given. You have to identify the statements whether correct or incorrect. In lack operon, the repressor prevents the binding of RNA polymerase to lac promoter. Absolutely true. So the repressor binds to the operator region and some portion of the repressor also binds to the 5' end of the promoter. If the repressor occupies even the promoter region, RNA polymerase cannot bind to the promoter and thereby transcription is prevented or transcription is inhibited. Prevents the binding of RNA polymerase to lac promoter. This is correct. First statement is correct. Statement 1 is correct. In lack operon, the inducer prevents the binding of repressor to operator. This is also correct. The inducer is lactose. More accurately, it is yellow lactose. So that yellow lactose binds to the repressor. Repressor has got two binding sites: operative binding site and inducer binding site, or the substrate binding site, yellow lactose binding site. So when the lactose or yellow lactose binds to the repressor, conformational change takes place in the repressor, and as a result, repressor is detached from the operator, or the repressor cannot attach to the operator. So The inducer prevents the binding of repressor to operator, where a repressor prevents the binding of RNA polymerase to the promoter. So both statements are correct. Both statement 1 and statement 2 are correct. So fourth option must be the correct one. Let us see the answer. Fourth one. In lac operon, the repressor binds to the operator and also to some region of the promoter to prevent the binding of RNA polymerase at promoter. The lac inducer, that is lactose, after being converted to L-lactose, binds to repressor, brings about conformational change in the repressor and the repressor either fail to bind to the operator or detach from the operator to promote transcription of cluster of structural genes, that is ZYA genes. Transcription is prevented. Next one. Choose the incorrect statement. Francois Jacob was biochemist. No, Jacob was a geneticist. This is incorrect one. Lac operon is repressible operon. No, lac operon is inducible operon. The repressible operon is that operon which is not expressed when an end product of a metabolism is present. For example, tryptophan. Tryptophan is the end product of the metabolism. When the tryptophan is present, then the operation of operon is repressed. Right? That is inhibited. The lac operon is inducible operon, it is not repressible operon. So this is also incorrect. Lac operon shows both negative and positive regulation of transcription initiation, absolutely true. But mainly it shows negative regulation. So negative regulation is due to the influence of the regulated protein, that is repressor protein. Repressor protein. Right? So product of gene I forms the repressor. The repressor binds to the operator, thereby preventing the binding of RNA polymerase to promoter. So that regulation where RNA polymerase is prevented to bind to the promoter by a repressor which binds to the operator, such type of regulation is referred as a negative regulation. Positive regulation means here other accessory proteins known as activators, they bind to the promoter, particularly 5' region of the promoter, and thereby enhance the binding of the RNA polymerase at the promoter site to initiate the transcription. So in case of lac operon, some positive regulation is also shown, but that is due to the catabolite activator protein. Upon binding with the cyclic AMP, that complex binds to the promoter, thereby enhances the binding of RNA polymerase. Right? So third option is the correct statement. Recessive mutations in LAC-Z gene can prevent the binding of RNA polymerase to the LAC promoter. No, it cannot. Any mutations occur in LAC-Z gene, so the product of the LAC-Z gene cannot be formed, but it cannot prevent the transcription or binding of RNA polymerase to the LAC promoter. Binding of RNA polymerase to LAC promoter is prevented by the repressor upon binding to the operator. Right? The third option must be the correct one. Let us see the answer. Lac operon shows both a negative and positive regulation. So, negative regulation by repressor, product of gene I. Positive regulation, another regulatory gene product, that is catabolite activator protein. This catabolite activator protein binds to cyclic AMP. Again, cyclic AMP is formed when there is negligible concentration of the glucose. But cyclic AMP is formed by the enzyme adenylate cyclase. So the cyclic AMP, the phosphodiester binding is between the fifth carbon of the sugar as well as the third carbon of the sugar. So that cyclic AMP is required for positive regulation or binding of catabolite activator protein to the 5' end of the promoter to enhance the binding of RNA polymerase to promote transcription or to initiate transcription. Francois Jacob was a geneticist, not biochemist. Lactoperon is inducible operon as the transcription is promoted in the presence of lactose. It is converted to low lactose. Excessive mutations lack Z gene can prevent the formation or low-level formation of beta-galactosidase but not binding of RNA polymerase to lac promoter. Coming to the next question. Assertion reason. A low level of expression of lactoperon takes place all the time in E. coli cell. Absolutely true. All the time a low level of expression of lactoperon takes place in E. coli cell because the repressor permanently not attached to the operator. So it attaches and now and then detaches from the operator. Whenever it detaches from the operator, some transcription of structural gene takes place. As a result, some amount of beta-galactosidase or few molecules of beta-galactosidase, few molecules of amylase and few molecules of trans-acetylase are found. The assertion is correct because the lac repressor periodically attaches and detaches from the lac operator in the absence of lac inducer. In the absence of lac inducers, it attaches and detaches, but much of the time attaches. Sometimes detaches. Whenever detached, RNA polymerase binds to the promoter and transcription is carried out. The assertion is correct, the reason is correct, and reason clearly explaining the assertion. So A and R are correct, and R is the correct explanation of A. So you should carefully note this one because the low level of expression lactoperon takes place all the time in E. coli cell is given in the book, but the reason is not mentioned why there is low level of expression of lactoperon. So you should be very careful. Second option must be the correct one. Let us see the answer. In the absence of lactose and also in the abundance of glucose, the lac repressor attaches most of the time and detaches for some time from the operator. Whenever detached or whenever detaches, RNA polymerase binds to the promoter and some transcription or translation of lac ZYA takes place. Right. Coming to the next question. Which one of the following is involved in switching on and off of the lactoperon? So, the switch is operated and it is kept in on position whenever there is lactose or low lactose, and whenever there is no lactose or low lactose, the repressor binds to the operator and operator switches at off position. So it is the lactose that keeps whether the operator switch kept in on position or off position. Lactose, not repressor. Repressor keeps it in off position. Beta-galactosidase is not at all involved in operating the operator switch. Trans-acetylase, it is also not involved. So third option must be the correct one. Let us see the answer. Further. Lactose is involved in switching on and off of the lactoperon. Whenever lactose is available, the operator switch is kept at on position by detaching the repressor from operator. When it is absent in a medium, the repressor is bound to the operator and lactoperon is switched off. Beta-galactosidase is involved in metabolizing the lactose and converting the lactose into allolactose. Repressor product of lac-I. Repressor is product of lac-I. It prevents the transcription of structural genes by RNA polymerase. Trans-acetylase is involved in detoxification of beta-galactosides and their excretion by carrying out their acylation. The trans-acetylase transfer the acetyl group from acetyl-CoA to the beta-galactosides so that the beta-galactosides can be excreted. Excess beta-galactosides are there excreted, right? Or they are made non-toxic. So here, what is the answer? Lactose is the answer. Coming to the next question. Identify the correct statements. Which among the 5 are correct statements? Among the A, B, C, D, E, which are the correct statements you have to identify. Yellow lactose is main inducer of lack of operon. Absolutely true. So, main inducer, actually the yellow lactose that binds to the repressor and prevents the repressor binding to operator or detaches the repressor from the operator. So, A is correct. When mutations occur in the region of lac I with amino acid sequence for operator binding site. If mutations occur in the lac I gene where information for the sequence of amino acid needed for binding to the operator are present, lac polycystron is continuously transcribed. Absolutely true. In that case, the repressor, though formed, cannot bind to the operator. When the repressor failed to bind to the operator, continuously the structural genes or polycystron is transcribed and translated. B is also correct. Repressor, beta galactosidase and permease have lactose binding sites. Yes. Repressor has got operator binding site and lactose binding site or yellow lactose binding site. Beta galactosidase also has got an active site where the lactose is bound and converted to glucose and galactose. It can also be converted to yellow lactose. So beta galactosidase can catalyze isomerization reaction as well as hydrolysis reaction, mainly hydrolysis. When lactose is converted to yellow lactose, it is isomerization. And permease also has got the lactose binding site so that the lactose is carried into the cytoplasm from the surrounding atmosphere in E. coli. So it is bound to the cell membrane, right? It's bound to the permease of the cell membrane lactose, and the permease carries the lactose into the cytoplasm of the bacterial cell. So repressor, beta galactosidase and permease, all the three have the lactose binding sites. LAX gene is used as a marker in microbial genetic engineering to select recombinant transformants. Absolutely true. So one of the selectable marker will be LAX gene. LAX gene has got some restriction sites. So whenever a gene is inserted in the LAX gene restriction sites, so LAX gene is inactivated and there is no formation of beta galactosidase. So blue-white selection in microbial genetic engineering is due to using LAX gene as marker. So this is also correct. Cyclic AMP receptor protein is involved, that is CRPR, catabolite activator protein. Cyclic AMP receptor protein is involved, right? It is also known as catabolite activator protein. Involved in positive regulation of lac operon transcription. Absolutely true. So cyclic AMP receptor protein binds to cyclic AMP and these together bind to the 5' region of the promoter, that is cap site. So thereby it enhances the binding of RNA polymerase to promoter and the polysystron structural genes are transcribed. So E is also correct. A correct, B correct, C correct, D correct, E is also correct. All the 5 are correct. So fourth option must be the correct answer. Let us see the answer. Allolactose is the main inducer of the lac operon. Only this one binds to the repressor to then detach it from operator. Mutations in operator binding site information region of lac I prevents the binding of repressor to operator and the polysystron will continuously express at higher levels. Lactose can bind to repressor, beta galactosidase, permease and transestylase. LAX gene is used as marker in microbial genetic engineering to select recombinant transformants from non-recombinants. Catabolite activator protein or cyclic AMP receptor protein can bind to cap site or promoter and helps in binding of RNA polymerase to promoter of lac operon to promote transcription. Coming to the next question. How long the lac operon would be expressed in the presence of lactose? This was the question asked in the lac operon on almost at penultimate paragraph. As long as lactose is available and glucose levels are low and immediately metabolize. Yes, first option must be the correct one. As long as levels of glucose and lactose are in equal concentrations, no. Glucose and lactose are in equal concentrations, the lactoperon is not expressed or poorly expressed. As long as repressor concentration is high and there is no cyclic AMP, no. The repressor concentration is high, repressor binds to the operator and cyclic AMP is not there, then catabolite activator protein cannot bind to the promoter. This is also wrong. As long as cap concentration, catabolite activator protein, right? Concentration is more than repressor concentration. More than repressor concentration. As long as CAP concentration is more, no, there should be cyclic AMP should also be present for binding of CAP, a catabolite activator protein to the promoter region of the structural genes of lactoperon. So the first one must be the correct one. Let us see the answer. First one. Lactoperon is expressed as long as lactose is in high concentrations. Glucose is not available. Cap and cyclic AMP are produced and promoter is not subjected to any mutations. Coming to the next question. Identify the correct sequence of various transcribed and untranscribed sequences of lactoperon. The correct sequence, lactoperon. So, actually the operon consists of the promoter, operator and the cluster of structural genes together known as operon. So, though it is in the book, even LAC-I is also mentioned as a part of lactoperon. It helps in regulating the expression of structural genes of lactoperon. It is not actually part of lactoperon. LAC promoter, absolutely true. LAC operator is next. 5' to 3' these are LAC promoter, LAC operator, LAC Z, LAC Y, LAC A. So ZYA genes. Right? So first option is the correct sequence. Let us see another one. LAC operator, no. LAC promoter first, not LAC operator. LAC promoter, LAC operator, LAC Y, no. LAC Z should come first prior to LAC Y. LAC promoter, LAC operator, LAC A. No, LAC A is at the last, nearer to the terminator sequences. Right. So the first option must be the correct one. The correct sequence is LAC promoter, LAC operator, LAC Z, LAC Y, LAC A. Please like, comment, subscribe, and share. Thank you very much.